So, moving on to chapter 10, we're skimming a kingdom. We're going to look at the protozoa, which are not the protists. The protists are the kingdom, the protozoa are a subset of the protists. We've already talked about the algae, and I mentioned that they were pushed into the protist kingdom. It says amoeba conventions, and those are the sort of little name tags they give you at some conventions where it just sticks on you, and they all have their own names, like Lola and Selin and Steve and Jack. Of course, they would not be males and females. Amoebas as a group very, very rarely engage in sexual reproduction, and when they do, it is not gendered. I shouldn't say that. I'm sure there's exceptions. There's always exceptions. This is a unicellular group. Now, one thing to keep in mind, I'm not sure I've mentioned this before, we've looked at unicellular groups before. In order for a cell to be active, to be engaging and interacting with its environment, it needs water around it, or water with stuff in it, so that things can go in and out through the cell membrane. So in order for protozoa to be active, in order for bacteria to be active, they need water surroundings. Like bacteria, like we talked about with funguses, is that these guys have got to be able to get by when the water isn't there. And so they will have variations. I mean, the things that live in the oceans, obviously not. Things that live in permanent fresh water, obviously not. But things that can dry out are only going to survive as a species if they can survive that and come back later. And there's a bunch of drying resistant forms, including spores. Remember I talked about spores? Spores, cysts, all sorts of different types of casings. Protozoa are kind of all over the map in terms of what they can and can't do and what they can and can't build. And so they, almost anything you can think about for a single eukaryotic cell to be able to do, you're gonna find protozoa that can do that, including associated with one another in sort of multicellular forms. Remember colonialism, unicellular colonialism is something you find in this group, where the individuals can live on their own, but that isn't the way you find them in nature. You find them associated in a group, and the members of the group are not all doing the same thing. They're doing different functions that contribute to the overall group survivability. We'll look at one of those in the lab, although it's just going to be a preserved form because I couldn't get live ones this year. They don't like to live very long anyway. These things swim, they crawl. When they crawl, they typically do it with pseudopods. Imagine you're new to microscopes, back when microscopes were a new thing, and you're looking at this pond water that you caught, and you're seeing on the slide, you're seeing something that you put it on there and it's kind of round, and then you see it put an extension out, and then flow into the extension. extension, comes back as it flows forward. Puts an extension out, flows into the extension. Puts an extension out, flows into the extension. So I go, well, it's crawling with extensions that it's almost using like feet. But they're not really feet. They're just sort of feet. That's what this means. Pseudopod means not really feet, but sort of feet. That is probably a scanning electron micrograph. Not entirely sure what they did to the amoeba in order to be able to get it into the scanning scope, but it does show the extensions of an amoeba. It shows the pseudopods. Amoebas are not the only ones that do that. And pseudopods are not just used for crawling. I'm going to move this to the side a bit, come back. These are all cell extensions that are technically pseudopods. There's the classic extend and crawl ones. Some have a casing, and the things they crawl with look more like octopus tentacles that they kind of move along with. There's something called radiolarians that have these very long, thin extensions that help them to float so that they stay away from the bottom, is that they live in the water. They don't really swim, but they've spread their weight out so effectively that they don't sink particularly either. And then some of them actually use pseudopods like a netting. And some of them use them almost like a lasso. I've got a friend who, the fellow I've mentioned, who does his research diving under the ice caps in Antarctica and taking samples off the bottom, is that there are protozoans that live there that are very effective predators. They sit on the bottom, and as stuff goes by, they reach up with pseudopods and grab them, like an octopus with tentacles, and pull them down. So these are a lot more diverse in both form and usefulness than just the old crawly things that you tend to think of with an amoeba. And they don't just show up in the amoeba group. There's also cilia and flagella. Now these are kind of interesting because they are homologous. They have the same core structure. They have a 9+2 arrangement of microtubules, something we'll talk about more in the second semester when we get into the molecular parts of cells, little bitty parts of cells. So they're built the same, but they have a bunch of functional differences so I'm going to make a little chart here. Cilia are smaller, flagella are larger. Now, as an introduction to biology, you may say, so what's the cutoff? What's the threshold where if it's below that, it's a cilium, and if it's above it, a flagellum? Which, a nice structured science would have that. We don't have that. Is that the threshold line is more of a gray area. And there's some big things that are considered cilia and some little things that are considered flagella that kind of fit other things on the list, but it's also whatever somebody decided they were looking at originally. They kind of gave it the name, and the name kind of stuck. Is that biologists don't tend to be quite as jargon obsessed as some of the other sciences, where this definition just means this, is because biology is a science full of exceptions anyway. So you try not to get too locked into things. I wrote that wrong. When you find a cell with cilia on it, there's always lots of them, hundreds, if not thousands of cilia. If you're looking at cells that have flagella, you almost never find more than a dozen on a particular cell. So fewer than 12 technically means 11 or less, but it really should be 12 or less. So, and if you're gonna find cells that have a whole bunch of flagella, you're gonna be looking in some pretty odd places. Like inside the guts of wood-eating insects. There's a weird group that have hundreds of flagella. They're these egg-shaped cells where the flagella are all attached to the top. If you've ever seen the Addams Family, they look like the protozoan version of Cousin It, except without the hat and the glasses. But that's unusual. It's very, very unusual to find more than a dozen flagella. Very often you find 1 or 2, 4 or 6. They often come even numbers, but that's not always true either. Cilia, it's not unusual for a cell to be covered with them. They sometimes have bands of them. There's lots and lots and lots and lots of cilia on a cell that has cilia. Is that cilia, because there's so many of them, they are smaller, but they're packed fairly close together, and they could conceivably tangle with each other. So they're very well coordinated. If you watch cilia across the surface, and you see this ripple as, if there's a row of cilia and they do this kind of stroke. Imagine my elbow. I can't make my arm flexible enough to do it, and I can't keep my elbow really in one place and show you. But it's more of a push stroke. And this row might be here in the stroke. The row behind it might be there in the stroke. The row behind it might be there in the stroke. This row over here might be, so that they not only don't get in each other's way, but they produce this ripple wave effect that's a pretty good propulsion ripple. Is that we use that to move stuff in our bodies as well, where we have cells in a layer and they move stuff with cilia across the surface. But it's a very effective swimming stroke, tends to produce, because there's so many of them working in a coordinated fashion, tends to produce a very smooth type of gliding motion, even though the cells sometimes spin. Flagella, even when you have multiple flagella, they don't really work at working together. They sort of are coordinated, but it's not that tightly coordinated. I once was watching a cell swim across the field out of pond water, and it had 2 flagella on it. One of them fell off. It could not adapt. All it could do was spin in one place with one flagellum working until it regenerated a replacement for the one. Why did the one fall off? I don't know. Is that it's apparently very odd, because when I tell this story to biologists, they go, "What do you mean it fell off?" And I said, "I don't know, it just dropped off and it was wiggled there for a while." And I watched the new one get regenerated. It took about 15, 20 minutes. And as it got more and more regenerated, instead of spinning, it started to spiral, but at least I could stay up with it. Until eventually it had a second full flagellum, and then like a 2 outboard motor boat, it could push itself along. Whereas with just 1 outboard motor going, it could just push itself around and around in a circle. Because flagella are bigger, they sometimes have modifications. Is that the flagellum may start at the front of a cell and parallel the cell, and part of the cell membrane may come out and attach to it. So that when the flagellum moves, it's moving a big fin. There are flagella that have combs on the end of them, and will actually filter feed using the combs. There's a whole bunch of modifications that you can get with a fairly large mobile extension that you'll see. Because cilia are smaller, there's not a lot to build on. The closest you get to an add-on with cilia, when I had the Hydra set up in the lab for people to look at, there were little guys scooting around on the Hydra that looked like they were little crawling bugs. Those were ciliated protozoans, and their cilia are modified where they take about 7 of them and they fuse them together at the tip. And then they move them literally like they were little insect legs that they can scurry around on a surface with. And that's not really an add-on to an individual psyllium, it's the closest they get to something like that. This is not as dependable. But as I mentioned before, typically cilia are pushing and withdrawing, and pushing and withdrawing, and pushing and withdrawing, and rows and rows and rows and rows of these things. Whereas flagella tend to spin like a propeller. So you see them from the side, they look like they're wiggling. But if they turn and come towards you, you can see that the flagellum's going around and around and around. They don't all move that way. That's why the usually is on here. But it's a typical motion for cilia and flagella. Still continuing on some of the features for this group. Autotrophs, self-feeders. That's literally what it means in Latin. Okay, it's on the screen, but it's not on your screen. Photosynthesis synthesis is that I don't know of any chemosynthetic protozoans. There probably are some out there. I've never heard of it, but I never say never with some of these groups. So autotrophs usually are photosynthetic. A lot of them cannot do that. They need their food fully made. And some of the autotrophs also will grab and eat things, as well as produce their own food photosynthetically. So sometimes you get both. As I said, all over the map here. When you're a single cell, you eat by ingesting material into a membrane bubble and then digesting and absorbing the nutrients out of the bubble. That's a food vacuole. In some of the more complex protozoans, there is what they call an oral groove. It's kind of like a little entry gullet. The cilia pull food particles down. As they go down the gullet, they kind of get tasted for viability as food. If they're just some sort of floating particle that's not digestible, it often will get kicked back out. Down at the bottom of the gullet, bubbles will form, food vacuoles will form, and they will actually follow a path through the cell, almost like it was going through a digestive system, and they're gradually getting digested and nutrients are being absorbed. And then when it gets to the end of its pathway, it comes up to the surface. So the bubble comes up to the surface and then flows onto the surface at a special structure that is readily available for that. So this isn't in a lot of them, but it's a weird one that I'll mention later. So I'm going to put the term up here. That special, almost trapdoor for getting rid of the stuff that's left over when you've digested, same way we have leftovers. They have leftovers. And cytoproct is essentially a cell anist. I mean, think of what a proctologist does. Cytoproct, same root word. When these were first discovered, and they were discovered to have nuclei, people wanted to put them into the multi-cell groups. Assign them to the animal kingdom, assign them to the plant kingdom, assign them to the fungus kingdom. Except they weren't cooperative. Is that the plants would work sometimes and be very much like animals. The animals would sometimes be very much like funguses. And there came a time where I think the multi-cell zoologists just got tired of listening to these people argue about what groups they belonged in. and said, why don't you guys go and take a room of your own and argue amongst yourselves. And so the unicellular eukaryotes became their own kingdom. I'm sure that it was more complex than that, but you get the basic idea. They don't work by the rules of the multi-celled groups, is that there's a lot of slop over in features from one group to another. You can find them doing all sorts of different things. When you put a cell into fresh water is pretty much 100% water. A cell has got a lot of stuff in it that is not water, so the water level inside a cell is never going to be equal to the water around the cell. There's always going to be more potentially entering than leaving, because there's more on the outside trying to get in than is on the inside trying to get out. Now, one way around this is to put the cell in a casing so that water can't enter because it can't get the cell any bigger than it is already. It's hit the casing and it can't generate. Remember, root pressure can move water up a plant. This can also expand a cell and potentially pop it. But a casing on the cell can prevent that. Plants have been fine with that. Fungus has been fine for that. Protozoa, some of them have the casings. Some of them don't. They need a certain amount of movable flexibility, like an amoeba, or some lightness to them. So they need to pump the water back out. Water comes in by osmosis, and in order to keep the cell from swelling up and popping, it's got to be pumped back out. That's a contractile vacuole. Fills up, fills up, fills up, squirts the water out. Fills up, fills up, fills up, squirts the water out. It's a very common feature in freshwater protozoans. Protozoans that live in the ocean are in a balance. So, the ocean water has a bunch of salt and stuff dissolved in it. So the water levels are not 100% like freshwater. They're down here, and cell content, water content, is pretty much equivalent. It's so they're in osmotic balance. They don't need to pump water in or out in order to be in balance. A lot of needs are purely done by things moving from high concentration to a low concentration. If you're consuming oxygen, because you're doing cell stuff and you're using aerobic respiration, the oxygen levels in the cell are dropping. So there's always going to be more oxygen headed in than is headed out, because it's getting used. The process is generating carbon dioxide. There's more inside the cell headed out than is outside the cell headed in. That's diffusion. Waste materials, same thing. So a tremendous amount of exchanges with the environment where stuff is being used or converted or being produced that needs to be gotten rid of is not actively moved in and out. It just goes on its own by diffusion. Let's see. You find basically every type of reproduction you can think of. You find sexual reproduction. You can find budding, like we talked about with yeast. You can find binary fission. You can find multiple fission. If you have a fish tank and you get something called ick, that's called Ichthyotherius. It's a ciliated parasite. And what happens is it embeds itself under a scale, and it grows, and it produces lots and lots and lots and lots of nuclei. Copies, asexual reproduction. And it may generate hundreds, if not 1,000 of them. And then it will, what's called fragment. Each nucleus will get its own surrounding membrane, and you go from having one big multinucleated cell to 1,000 little ones that settle down into the gravel and grow a little bit, and then they go up and invade the fish. So this can very quickly, in a matter of a day or two, go from a couple of spots on the fish to being completely out of hand and killing the fish in the tank. So fragmentation is a possibility. Binary fission, where you divide in relatively equal amounts. Budding, where a small growth gets its own copy nucleus and can grow to be its own separate cell. Lots and lots and lots of different types of asexual reproduction. Sexual reproduction can involve genders with roles that vary depending on conditions. Very often it involves something called conjugation, which is just a haploid nuclei are produced and exchanged between partners. So there are no roles. They're both doing the same thing. So that's called a genderless sexual exchange. There's a lot of ciliated protozoans do that. The process gets very complicated. It sounds simple, but it's very complicated. We'll talk about that when we talk about ciliates. And one whole group, subgroup within the protozoans, does that back and forth, sexual, asexual, sexual, asexual. Very often with male-female genders, which when you think about it, in a single-celled organism is a little tricky, because what produces male, female, and multicelled? Sperm and eggs, forming the hold on to the food role and the seeker role. In apicomplexans, some of them become the let's build up some food, and some of them become I'm coming to find you. And so you do get male-female genders where the whole animal becomes essentially the sperm or the egg cell. Tricky. Okay, we'll do the subgroups next.